My name is Olivia Corradin. I am an assistant professor at the Department of Biology here at MIT and a member of Whitehead Institute. I came here first as a fellow in the Whitehead Fellows Program, which is an opportunity to start a lab right out of grad school. So started there and then recently was promoted to assistant professor. Before that, I grew up in Michigan and stayed relatively close to home to go to school. I went to Marquette University for my undergrad and Case Western Reserve University in Cleveland for my PhD. My lab is primarily a human genetics lab. We're really interested in understanding how genetic variants contribute to your likelihood of developing disease. I think often we think about genetic diseases as if you have this mutation, it is going to be a determinant, it's going to cause a disease. What we study are more like risk factors. So just like diet and exercise are types of lifestyle risk factors, genetics can be a risk factor as well. And understanding the genetics themselves as a way of understanding the disease and how it works within the body. So if we identify a genetic change that puts you at greater risk for developing disease, then we can ask, well, what does that genetic variant do to a cell? How does that cell influence a system? And that's how we can start to understand how the disease actually works. And so that type of study has been really prevalent in the last decade or so. Thousands of these genetic studies have been performed on any human trait you can think of. So things like schizophrenia, multiple sclerosis, but also like coffee consumption, whether you're a morning person or an evening person. All of these types of phenotypes have been studied in the genetic space. And this has just produced an absurd amount of data. We have identified as a field well over 100,000 different genetic variants that change your likelihood of developing these traits. And so what my lab focuses on is how do we use that information to understand the phenotype or the disease. And what complicated that pipeline of we've identified a variant, now we want disease insights, is that these variants are rarely inside genes themselves. So when we think about the area of your genome that makes genes, which make proteins, that's only about 2% of your total genome of all of your DNA. And so most of the genetic risk factors for disease are not in these proteins at all. Instead, they're in regions that help control which proteins are turned on and off in different contexts. So I like to describe this in the frame of what my sister studies. So my sister is an architect. I always tell her it's the equivalent of taking one blueprint, giving it to 3 contractors and having them return completely different buildings, right? Every cell in your body has the same genome, but yet builds a completely different function. So how does a neuron become a neuron and not a beating heart cell? That is all described by how genes are regulated. And so when you disrupt this regulation, that is often a contributor to human disease. And so that's where our major focus is at.